Thank you very much for that nice introduction and for the invitation to speak today. I'm excited to be here to share some new but ongoing work that I've been doing on epigenetics. So epigenetics is a hot new field of post-genomic science that's offering a really new and dynamic understanding of the relationship between environments, genes, bodies, and health. And emerging is an entirely new and non-determinist model of biological life as plastic, that is, as changeable. So this talk is taking off from some ideas about epigenetics that I started to develop in earlier publications, including with Julie Guthman here in the front, and her ideas have influenced this paper also, and so I definitely wanted to acknowledge her contribution. Thank you, Julie. And here I'm extending from our earlier work by doing a critical analysis of the scientific literature itself to really understand how this new dynamic understanding of bodies and environments is developing. And as we go, you're going to see a lot of quotes and figures from scientific papers, starting with this one from Science. And so I'm going to work through some of this actual scientific literature with you. I know some of you, it's not something you read regularly, but we'll try to make it interesting as we go. So epigenetic mechanisms are those that are defined as those that influence not genetic sequences themselves, but how genes are expressed. And these are chemical and structural changes to how genes are packaged within our cells, which influence which genes are then available to protein-building RNA. Environmental epigenetics is a field which studies how environment, which is broadly defined to include everything from toxic exposures to nutrition to social interactions, affects these cellular mechanisms and therefore how these epigenetic signals influence kind of bodily outcomes. So in this figure, we have environmental factors, which are on the top there, stress, diet, epigenome, contaminants, interacting with genes from the parents, the parental genome on the bottom, on the left there, and they combine to influence individual development, and then influences current predisposition and future generational disposition to health and disease. So my question is about how this new model of biological plasticity is affecting concepts of difference, and in particular, notions of normality and abnormality. So this talk is about how epigenetics is contributing to a new biological yet non-determinist ontology of race, and about how the fetus, seen here, and reproductive women are emerging as central figures in this new epigenetic biopolitics of race and bodily plasticity. I thought I wrote this without the word biopolitics, but there it is. So my point is not how this new science, I just want to emphasize this, I'm not really talking about how this new science is taken up by others, by policymakers, the press, activists, multiple publics, but how questions of abnormality, race, and reproduction are shot through the science itself. So I'm really kind of staying inside the science here and thinking about that in terms of truth-telling and in terms of governance, both. So environmental epigenetics, I think, really offers much that should resonate with geographers. It is a science of variation, a science of difference, again, that takes us away from determinism and individualism and toward a notion of biological difference as part of the warp and weave of both space and time, both social and natural. So that what happens inside of us, even at the molecular level, is entirely dependent on what happens outside of us, including in the past. So by focusing on the ways that the environments, multiple environments, interact with genetics to shape bodily outcomes, epigenetics is inherently spatiotemporal and socio-natural. Like social theory, epigenetics blurs or even obliterates these kind of dualistic distinctions that we've been grappling with for decades now. So epigenetics is also appealing because it lends itself to arguments for social and environmental justice. For one, it provides a mechanism for how environmental exposures make biological differences. And this is really important advance in the face of ongoing denials, for example, that environmental chemicals have biological effects. So we can see that they do. It also shifts the focus away from the individual and how their genes or behavior create their bodily outcomes. Instead, it focuses our attention on the broader environment. It suggests collective causation for and responsibility for broad health outcomes. And in doing all of this, it also appears as an anti-racist science, and this is emerging in the literature itself. Certainly it undermines our genetically determinist notions that underpin modern race and racism, But even more, it does this in a way that shows how racism produces biological difference, how racism can produce race. And I will be coming back to this as we go. So for all these reasons, environmental epigenetics seems to be the life science for which many of us have been waiting. Yet, always, of course, the yet. Looking to the science as it's actually unfolding, we can also see, and maybe I use unfolding as a pun here for life, we can also see that this non-determinist approach to bodily variation, the science of plasticity, contains within it a very different and much more troubling set of possibilities. And indeed, I will demonstrate that epigenetics seems also to be the science of a new eugenics. This ugly side of epigenetics arises out of the heart of what makes epigenetics promising, that it focuses on plasticity rather than determinism. The plasticity of our spatio-temporal socio-natural life makes it open to intervention, but then intervention is premised on notions of abnormality and normality, or even optimality. And I'm going to demonstrate this in 4 steps. So first, I'm going to show that epigenetics is the science of variation. It shows us that biological difference is indeed inevitable, and that therefore there is no normal. So here I'm going to focus on the very wide-ranging environmental factors that are identified in epigenetic science. Second, I will show that, perhaps paradoxically, epigenetics is also the science of disruption, and it expands our notions of abnormal. And here I'm going to focus on the wide-ranging outcomes that are studied in epigenetic science, demonstrating that epigenetics actually radically expands notions of disease to encompass an enormous range of conditions. Third, I will show that environmental epigenetics not only expands our notions of abnormal biological differences, but it identifies ways to eliminate these differences. And here I focus on the scientific search for epigenetic therapies to cure epigenetic abnormalities. And fourth, I show that epigenetics is a reproductive science, and I focus on how the uterine environment is figured as a key time space of epigenetic becoming. So in short, my argument is that in this focus on abnormality and optimization, epigenetics is tied to a eugenic logic even as it rejects notions of genetic determinism. That is, epigenetics not only ties difference to the biological body through these molecular mechanisms, but it uses those molecular mechanisms to diagnose abnormalities and seeks to intervene in these molecular mechanisms to eliminate those variations deemed abnormal. In other words, it might seem that new epigenetic models of plastic life should eliminate race by eliminating notions of discrete kinds that are given in nature. Instead, it appears that epigenetics offers a new form of racialization based on processes of becoming, and it seeks to control processes of becoming by controlling reproduction. So to provide some very brief background, I'm going to present 3 very simplified models, genomic models of life for explaining individual traits and biological variation. Simplified is definitely the word. So first is genetic determinism. Simple. Here, basic traits from physical characteristics to intelligence to many diseases are fundamentally genetic. They're given in our DNA. There's a unidirectional flow of information from DNA outward to proteins and to the organism, and even for sociobiologists to society. Life circumstances are not entirely inconsequential, but they make a relatively minor difference within a narrow window of possibility that's provided by our genes. And of course, the environment is important for evolution, but environment in evolution, in an evolutionary sense, is a passive selector. It doesn't come into the body in any meaningful sort of way. And it's this notion of genetic determinism that underpins much genomic medicine, which searches for and seeks to cure the bad genes thought to cause diseases. And of course, genetic determinism also underpins modern racism, in which races are considered to be biological kinds with different genetic underpinnings, and inferior races have bad abnormal genes. But findings in genomic science also undermine genetic determinism. And so I'm not going to focus on social theoretical debates here, but really within genomic science. And certainly the limited success so far of genomic medicine has suggested that genetic causality is much more complex than was expected by many. Also, efforts to map the human genome, completed in 2001, yielded a profound challenge to ideas of biological race. there's just as much variation within our socially defined racial categories as there is between them. Genetic difference does not map onto socially signified races. So how does genomic science then address these outcomes? It suggests that genes don't map really simply onto socially relevant categories, whether of race or disease. One way has been to take seriously this idea that nurture, our life circumstances, matters more than is presumed in genetic determinism. So one such kind of post-genomic model is known as gene-environment interactions. And in this model, environment is crucial, but DNA mediates biological response to environmental stimuli. It almost acts as a filter. Environmental factors pass through genetic inheritance to determine bodily response. So environment isn't shaping DNA so much as it's going through it to shape, and both of them together shape bodily traits. So given the same environmental stimuli, we would expect different responses due to different genetic characteristics, which might help explain why people with the same exposures to toxic chemicals have different outcomes. They have different genes. Now this is certainly a different model than genetic determinism, but it seems to leave intact, actually, I think, the underlying debate, which is about the balance of nature and nurture in shaping our biological destinies. For example, this model could be used to show how race as genetic difference affects susceptibility to environmental exposures, as I just said. So it leaves intact could leave intact some of these genetic notions of race. Now, epigenetics does something quite new. It still definitely focuses on interactions between genes and environment, but it does so in this very different way. Genes do not act as mediators between the environment and bodily states. Instead, epigenetic mechanisms act as mediators between the environment and genes. So these things happening inside about the ways in which genes are packaged is the mediator between environment and genes. And these are not fixed mediators, but are themselves quite dynamic, influenced by ongoing interactions. So both environment and genes are causal factors, and bodily states, including epigenetic marks, are emergent outcomes. So a very different kind of model. And this is how we get the idea that racism actually produces race. Racism leads to all sorts of differential environmental exposures that create biological differences between socially signified groups of people. So socially signified groups of people are treated differently, they get different environmental exposures, and that would produce then biological difference between these groups. So this is a very different understanding of race as an emergent outcome.